Hello, this is Mistaken Ford, and we're going to continue our discussion with DNA by looking at how DNA replicates. But we can't do this without understanding the history of how they discovered it. It's a rather fascinating one, and it does require a bit of background to kind of understand the logic of the experiment, but once you realize it, it's a really neat experiment. Where we left off the other video, we were just talking about the significance of the double helix model that Watson and Crick outlined, and their model very easily gave this idea of how DNA could be replicated. If you separate it out, you have the two parent templates right here, and then you can add on the new strands based on what the template is saying. However, that hadn't been proven yet. And so along comes two scientists, Meselson and Stahl. And Meselson and Stahl in 1958, they did a really neat experiment called the pulse chase. Now, we've actually seen an experiment already with the pulse chase, and that was about a month and a half ago. We did the pulse chase experiment with the labeled amino acids for the rough ER making proteins, and then watching where the proteins go with the cells. And we were looking at that with regard to where those proteins could be shipped in the cell. And that really showed that the Golgi body and the rough ER were really pivotal in directing the traffic of the proteins. So this is a similar experiment where they used an isotope. In this case, it's not radioactive. It's just heavy. It's a heavier radioactive. So they used nitrogen-15. It's an isotope of nitrogen. Nitrogen normally has an atomic number of 14, so it has an extra neutron. So first we need to understand that there were three competitive models of how DNA might replicate. The conservative model stated that when a DNA strand replicates, the parent strand will ultimately stay the same, and then you have the new strand being made entirely of new nucleotides. And then you had the semi-conservative. And the semi-conservative, you have the two old strands, and then the new strands each get one old and one new. So here we have the old and then the new, the old and then the new. And then they did have one where they couldn't quite determine a mechanism for this, but it had to be considered. And that was called dispersive, where the old parent strand disperses throughout the new ones. And in this case, you can see that you have old and new and old and new. So what they did is they grew bacteria using nitrogen-15. And because DNA has quite a lot of nitrogen in it, what you ended up getting were bacteria cells that were slightly heavier. Now, if you spin the bacteria in a centrifuge, and they're in a tube, in a test tube, let me try to get the same color here. Because they're heavier, the bacteria will settle in the centrifuge a little bit lower than if that bacteria were grown in normal nitrogen. So this would be normal, and then we go back to our heavy, and because it is slightly heavier, it'll actually sink when spun down. So they actually ended up with an interesting way to measure whether or not it had a heavy or a light nitrogen. So before we get to the experiment or the mechanics of the experiment, let's think about like how this could be. In a conservative model of replication, if you have an original, so this would be the original, If you had an original DNA strand, or we call it parental, and we grew it using N15 and it's heavier, what will happen if we take this heavy DNA in a bacteria and then transfer it to a culture of regular N15? So imagine, if you will, you're building a house and you're using a dark brick, and suddenly you switch bricks on the construction builders, and you would notice that the house is much darker at the bottom of the foundation. And then as they build the house, it turns lighter. Well, if you have that idea in your mind, that's exactly what they did. They transferred the bacteria to the normal nitrogen, N14. And so let's follow the logic. If, and that's a big if. So if it's conservative, then think about what we'd see. We'd see the original strand with two heavy nitrogen strands. Once they replicate, we would see a light form and then a heavy form. And of course, these would be the originals. And if that happens, and so the first round, we'd see one group of light and one group of heavy. But let's now compare. What if it's semi-conservative? If it's semi-conservative, we would start out with the original, the parental strands that were heavy, but notice the difference with one replication. Suddenly we have the original heavy and the original light in both strands. Now think about what that would do if we spin it out. In the original, what would happen in the first replication? In the first replication, we would indeed get one heavy and then one light band, as this pulls down heavier and then this pulls down slightly above. However, in the semi-conservative model, in the test tube, if we just spun it, we would just see a mixture smack dab in the middle of where the light and heavy would be. We would just see one band after one round of replication. And then, and in fact, they did this. So notice how wonderfully predictive this is. They have an if-then. They have a prediction. If it's conservative, we should see two bands. If it's semi-conservative, we should only see one band. So let me show you what they found. This is the actual results. And they could actually watch how everything's separated. So this is the start. So notice the line here, increasing density. And so this means it's way down here. This is the heavy. And so the replication continues, and notice as the replication is continuing, the band starts to shift up, and then notice this is generation 1, which means this is at the end of the first replication. Lo and behold, we only have one band. So rather a nice challenge. We know exactly, now we know. We can eliminate conservative replication. And so in the first round, the conservatives knocked out. It is not conservative. So, oops, I spelled that conservative. And however, we do have dispersive, and dispersive was a bit of a conundrum, because dispersive, you would get a similar look in the first round. You would get it kind of right in the middle with the dispersive. So what they had to do is they had to think, okay, well, what would happen in the next generation? And in the next generation, continue the logic. This would separate out into you would get one strand with the old and new, and then with the light one, you get two light Light and light. So again, imagine if we keep building the brick house, but this time the wall, the house is divided, and you'd see the house with some the darker bricks, but then we see the part of the house that only has the light. And so we get a difference here. So in the second round of replication, what we would see is that same mix, that hybrid a little above, but then we'd see a round of DNA that is only light, and so it's slightly higher. So indeed we get two bands. But notice in the dispersive, so follow what happens with the colors. In the dispersive, we would get a mix of light and heavy all spread out. And so the prediction would be in dispersive, we would see the bands just slightly higher, but we'd only see one band. So now again, we've got an if-then statement that allows us to conclusively say, if it's semi-conservative, it should look like this. If it is dispersive, it should look like this. And notice in the second round, lo and behold, we have two nice bands that kind of show exactly what happens. And you can even go, okay, here's the third round, and we see still a little tiny bit of the heavy, but mostly light, and it just edges up and up and up and up, and then they were able to conclude more and more conclusively that that's what it was. And we're going to take a look at this picture in class tomorrow. This is a nice one that kind of shows how they measured it, and you can see the bands changing. So thanks. Hope this helps, and we're going to take a look at this tomorrow in class.